good morning or good afternoon, depending where you are right now. My name is Henver. I'll be hosting our journal club today with Magnus Larsen and Dr. Henver Brunetta. So it's a pleasure to be here, and I would like to start by thank you the Journal of Physiology and the Physiological Society for organizing this virtual journal club. Since we're not able to travel right now for a conference, this is a great opportunity to be with our friends, to be with our colleagues, and discuss science as well. So thank you very much. And I'd like to thank you also, make a special thank you to Rosie, who has been nothing but awesome to now in organizing this webinar. Okay. So today there will be three presenters. Me, Henver Brunetta. I have a PhD in physiological science from the Federal University of Santa Catarina in Brazil, and right now I am a postdoctoral fellowship at the University of Campinas in Brazil under the supervision of Dr. Marcelo Mori. My research is focused on mitochondrial physiology and metabolism, so I'm interested in studying mitochondrial physiology in peripheral tissues such as skeletal muscle, adipose tissue, and heart in several conditions like obesity and exercise. Magnus Larsen is the first author of this paper we're going to discuss today. Magnus is from the University of Copenhagen. I'll introduce him shortly right before he will present his data. So thank you again, Magnus, to be here. And Dr. Henver Brunetta from the University of Bout, which is the editor, is member of the editor board in the Journal of Physiology. So thank you again. Please feel free to share your thoughts about the virtual journal club meeting using the hashtag #JPJournalClub on Twitter. Don't forget to mention the Physiological Society, Journal of Physiology, me, and Dr. Henver Brunetta. Okay, before we get started, some information. This meeting will be recorded and emailed to you, so you receive an email with this meeting in a week. So feel free to share with your friends and colleagues who couldn't be here today. And at the end of this meeting, there will be a Q&A section with Magnus and Javier. So feel free to make all the questions you want during the section. You can press the button at the bottom of your screen and make the question. You can do that anonymously if you want, but if you could write down your name and affiliation, that would be great. So feel free to make all the comments you want and all the questions, and at the end, we'll be answering them. Also, there is a vote button, but if you want, so you can vote the questions, and those questions will be on top of the list, so I'll ask them first. So if you see some questions you like, just press the up vote. And last, there will be a network section after this webinar. So Rose will send the link for this network session, and we'll be in a different room, so when we can continue to discuss this paper and we can talk more about science and the findings. So this will be like a beer section in a real conference, which no one would miss. And so feel free to join us in this network session. It will be really great to see everyone and to talk more about Magnus data and science. Okay? Just a little bit more information about how to get involved with the Journal of Physiology. Basically, there are three options you can use to be involved. The first one is to host a future meeting of the Journal of Physiology virtual journal club, which I'm doing right now. So this virtual journal club is the first one of 2021, but every other Wednesday, Journal of Physiology has a different virtual journal club, so you can be a host. It's a great opportunity to see your colleagues, meet new people, and to learn more. The second one is to write a journal club article on a recently published article in the Journal of Physiology. So there is a section in the Journal of Physiology where you can write a journal club article. And the last one is submitting your research to the journal. So Journal of Physiology publishes original papers in all areas of physiology. So if you have, please contact Journal of Physiology and they will be happy to answer you. Okay? So let's get started. Why did I choose this paper? So the title of this paper is the insulin sensitizing effect of a single exercise bolt is similar in type 1 and type 2 human muscle fibers. Magnus Larsen shared the first authorship with Dr. Steenberg, and the research group is located in the University of Copenhagen. So exercise is known to have numerous positive health effects and is a very interesting tool in prevention of chronic disease. So we can list at least 30 or 40 chronic disease that chronic exercise has a positive effect. Regular exercise results in a number of adaptive responses that contribute to positive health benefits. Although many of these adaptations are observed in skeletal muscle, primarily, exercise also has positive effects in central nervous system, adipose tissue, cardiovascular system, and liver. Okay, so I believe the paper from Magnus and his colleagues has several interesting findings, and we can discuss today, mainly about the adaptations regarding skeletal muscle. So today our focus will be on skeletal muscle, but keep in mind that chronic exercise has effects on several different tissues. When you talk about metabolism, chronic exercise is able to improve tolerance, insulin sensitivity, and metabolic flexibility. So this improvement in whole body glucose tolerance and insulin sensitivity is a good way to prevent type 2 diabetes. So an improvement in insulin sensitivity decreases the necessity of production of insulin from the beta cells, which can delay and or prevent the appearance to diabetes. Here I showed resistance training or endurance training. So both of types of exercise are able to improve metabolism, although we can see differences between them. So keep in mind that there are differences between the physiological adaptations regarding the type of exercise. But both of them are able to improve metabolism and insulin sensitivity. Also, we can see an improvement in mitochondrial content and function, as well as immunity. So for quite a long time, we know that exercise can improve metabolism. But here we talk about chronic effects of exercise. But we have to keep in mind that the physiological adaptations of exercise are a result of multiple single sections of exercise, and those single sections of exercise produce a lot of signals that contribute to this chronic effect of exercise. Among those signals, I'd like to highlight alterations in calcium handling, for example, activation on Kiens B, that we can see during exercise. Energetic status of the cell, for example, alteration in ATP and P ratio that occurs during exercise, and redox balance by the of relative oxygen species. So these three not only these three, but I think the main signals are involved in the chronic effect, chronic physiological adaptation. Okay, so during one section of exercise, the increasing metabolic rate results in the mobilization of different substrates to support ATP resynthesis. So the oxidative metabolism of carbohydrates and fat provides almost all the ATP for contracting skeletal muscle cells. The major substrates are muscle glycogen, as well as blood glucose derived from liver glycogenolysis or gluconeogenesis during exercise. Also, we have fatty acids derived from skeletal muscle or from adipose tissue stores. So we rely a lot on glucose and fat during exercise. But also, it's important to remember that carbohydrate oxidation, particularly from muscle glycogen, dominates at higher exercise intensity. So here you can see intensity above or around 85% of VO2 max, a strong reliance on glycogen, muscle glycogen. But on lower intensities, we rely more on fat oxidation to fuel our exercise. Okay, so that being said, I'd like to congratulate Magnus and his colleagues for this paper. I'm looking forward to hear your thoughts. Magnus is a master's student in human biology at the University of Copenhagen, under the supervision of Dr. Jorgen Wojtaszewski. And thank you very much, Magnus, again. Congratulations, and the stage is yours.
Yeah, thank you, Henver. Okay, so yeah, first of all, thank you, Henver, for that nice introduction and for choosing this paper for the topic of this meeting. I am very honored about the opportunity to present this work here, so thank you for the invitation. Obviously, what I will present for you is our paper titled "The Insulin Sensitizing Effect of a Single Exercise Bout is Similar in Type 1 and Type 2 Human Muscle Fibers." But before I tell you about the study, I do want to acknowledge all the work done by the co-authors listed here. Their effort has really been helpful throughout the process of making this study. So the idea for this study is based upon the insulin sensitizing effect of exercise, which is schematically shown in the figure you see here. So here you may notice that during an exercise bout, glucose uptake increases in exercising muscle. And after the exercise bout, this enhanced muscle glucose uptake again returns to resting levels within a couple of hours. However, with the following insulin stimulation, as obviously occurs during intake of food, then the insulin action is potentiated in the prior exercise muscle, leading to a markedly higher glucose uptake compared to rested muscle. And so we believe the physiological advantage of this enhanced glucose uptake might relate to a faster buildup of glycogen. However, the reason we are really interested in this phenomenon is due to the potential of pharmacologically targeting this effect and thereby creating a very potent insulin sensitizing drug that could be used in future treatment of diabetes. However, in order to design such a drug, we have to know the underlying molecular mechanisms. And although not fully elucidated yet, studies so far have led us to appreciate that a lot of this exercise-induced effect is due to regulatory mechanisms within the muscle fibers. So an intracellular event as a major contributor to this enhanced muscle glucose uptake. Furthermore, then studies in mice have shown that this specific protein, TBC1D4, is functioning as a point of convergence between exercise and insulin-induced stimuli towards enhancing glucose uptake. And to show you a bit more detail about this specific regulatory mechanism, then what we currently believe is going on is a mechanism that is induced during exercise. Because here we know that the enzyme AMPK is highly activated. And that goes especially for the AMPK complexes containing this gamma-3 subunit. Now this complex, when active, can phosphorylate TBC1D4 at the site serine 704. And we know in humans that this phosphorylation is maintained several hours into the recovery from exercise. And from the studies in mice, we believe this specific phosphorylation have a potentiating effect on the insulin-mediated regulation of TBC1D4 and thereby contributing to the enhanced glucose uptake in prior exercised muscle. And so several human studies have already evaluated this specific regulatory mechanism in regard to the insulin sensitizing effect of exercise. However, so far, all of the analyses made have been conducted in samples from whole muscle biopsies. And as well known, skeletal muscle consists of different fibers, which can overall be divided into two different types, type 1 and type 2 muscle fibers. And for this study, we wondered whether there was a difference in the insulin sensitizing effect between these two fiber types, thereby making one of them the primary driver of the insulin sensitizing effect of exercise in skeletal muscle. The reason we got this idea was due to a previous study conducted in our group showing that the AMPK gamma-3 subunit is markedly higher expressed in type 2 fibers compared with type 1. And furthermore, during an exercise bout, phosphorylation of TBC1D4, serine 704, were increased to a higher extent in type 2 fibers. And that was even shown for two different types of exercise. So giving the higher expression of AMPK gamma-3 and the higher phosphorylation of serine 704 during exercise, it led us to hypothesize that the insulin sensitizing effect of exercise would be greater in human type 2 fibers. However, as the title of the paper reveals, then this turned out not to be the case. But nevertheless, that was our initial idea for actually conducting this study. So in order to perform this analysis, we utilized muscle samples from a previous study conducted by co-first author Dr. Steenberg. In this study, 9 healthy males were included. They were all young and fairly inactive. And these subjects performed an experimental protocol consisting of 1 hour of one-legged exercise. Then they rested for 4 hours before they had muscle biopsies taken in both the prior rested and the prior exercised leg. Then the subjects underwent a 2-hour euglycemic hyperinsulinemic clamp and again had biopsies taken in both legs. And so this experimental setup allows us to investigate the underlying signaling mechanisms of the insulin sensitizing effect of exercise. However, in order to do it in a fiber type specific manner, we had to prepare fiber type specific samples. The way we do that is by taking a freeze-dried piece of muscle biopsy and under a light microscope dissect it into fragments of single fibers. Then each single fiber will be dissolved in a sample buffer, and with the help of myosin heavy chain antibodies, we will determine the fiber type of this specific single fiber sample. Then we will collect all the type 1 determined single fiber samples and pool them into one big type 1 sample. And similar goes for the type 2 determined samples. So for one muscle biopsy, we will end up with 2 fiber type specific samples. Then we will repeat this procedure again with a new muscle biopsy that we want to include. And once we have done that for all the muscle biopsies, we at first control the purity of these fiber type specific samples before we also determine the protein concentration in order to align them all. And once we have completed this step, our fiber type specific samples are now ready for further signaling analysis by the use of Western blotting. The first thing we then wanted to ensure was that both fiber types were recruited during the exercise bout. So we looked at the glycogen content and found that it was markedly decreased in the prior exercise leg in both the type 1 and the type 2 fibers, and even decreased to comparable levels. So overall, that indicates that both fiber types were recruited during the exercise bout. Furthermore, we also wanted to look at insulin signaling induced during the insulin clamp. So we looked at phosphorylation of AKT, which is upstream of TBC1D4 in the insulin signaling cascade. And so here we found that the insulin clamp induced a similar phosphorylation of AKT between the two fiber types, and so overall indicating a similar insulin signaling. So by showing that both fiber types were recruited during the exercise bout and that the insulin clamp induced a similar signaling in both fiber types, we thereby ensured that both fiber types had the same initial conditions for inducing an insulin sensitizing effect of exercise. And whether that also would come out as a similar sensitization, we could now evaluate by looking at phosphorylation of TBC1D4. And more specifically, we wanted to assess the phosphorylation of this specific site, serine 704. And so here we found, starting in type 1 fibers, that 4 hours after the exercise bout, the phosphorylation level in the prior exercise leg were markedly enhanced compared to the rested leg. And furthermore, then this phosphorylation level in the prior exercise leg were maintained during the following insulin clamp. A similar regulation was found in the type 2 fibers, which also had an increased phosphorylation in the prior exercise leg and also maintained this phosphorylation during the insulin clamp. So overall, this at least indicates a similar pattern of regulation between the two fiber types. However, in order to directly compare the response between the two fiber types, we also performed analysis only on the phosphorylation data in the prior exercise leg in order to directly compare the response between the two fiber types. But here we found no significant difference between the two fiber types, neither before or after the insulin clamp. And so overall indicating that serine 704 were regulated to a similar extent in both fiber types in the prior exercise leg. To support this further, we also looked at other phosphorylation sites on TBC1D4 and overall found a similar phosphorylation pattern between the two fiber types. Now, as expected from previous studies, we did find a higher expression of both TBC1D4 and AMPK gamma-3 proteins in the type 2 fibers. However, as you see from the phosphorylation data, then as long as the phosphorylation is related to the total amount of TBC1D4 present within the specific fiber type sample, then the overall phosphorylation pattern do not seem to differ between the two fiber types. And so overall, we interpret these data as a similar signaling towards enhancing glucose uptake in both fiber types. And here it would have been ideal with a measure for the eventual biological outcome, which would be enhanced glucose uptake. However, in this current fiber type specific analysis, it is not possible to measure glucose uptake. So as an alternative marker for how much glucose were actually coming into each fiber type, we chose to look at glycogen synthesis in the prior exercise leg during the insulin clamp. And although expecting this amount to be very small during a 2-hour insulin clamp, then we could actually detect at least a tendency towards an increased glycogen content after the insulin clamp. However, there were no significant difference in the response between the two fiber types, and so overall indicating a similar glycogen synthesis in both fiber types. To support this, we also looked at signaling towards glycogen synthesis by looking at phosphorylation of this rate-limiting enzyme, glycogen synthase. And so here we found in type 1 fibers that the phosphorylation level in the prior exercise leg were markedly decreased compared to the rested leg. and thereby indicating activation of glycogen synthase in the prior exercise leg. Furthermore, then this phosphorylation level were further decreased in the exercise leg after the insulin clamp, and thereby indicating further activation. And this regulatory pattern was similarly found in the type 2 fibers, and therefore overall supporting that there was a similar glycogen synthesis in both fiber types. So that led us to overall conclude that in recovery from exercise in healthy subjects, insulin regulate key proteins in glucose metabolism, similarly in both fiber types. And so we interpret our findings as that both type 1 and type 2 muscle fibers contribute to the insulin sensitizing effect of exercise in humans. And so if a pharmacological compound would be developed targeting this insulin sensitizing effect, then we believe our data indicate that both type 1 and type 2 fibers could be potential targets for inducing this effect. And so with that, I would like to thank you all for your attention. Thank you.
And so I think now I'll describe, so I'm Javier Gonzalez, by the way. I'm one of the editors who handled the paper, and I'll just describe very briefly why we thought this paper was worthy of publication in the Journal of Physiology. So whenever we get a paper submitted to the journal, there's a few things we're looking for. We accept papers in all areas of physiology and pathophysiology. We're particularly keen on clinical or translational focus to help further our understanding of the role that physiology plays in health and disease. And we particularly prioritize original work that illustrates new physiological insights or mechanisms. And I have to say, when we're evaluating these papers, we're indebted to the expert reviewers who provide their comments and insights. So we're really grateful for the time and effort that the reviewers put into high quality reviews. So one of the things we look for is, does it fit within the scope of the journal? And clearly with this paper, we felt it did. It shows in humans some physiological insights into this fiber type specific response to exercise and insulin stimulation. And it uses a novel method that was explained very clearly in the paper. There's a nice schematic that was shown there as figure 1. Is the question important for the field? It certainly advances our understanding. It was highlighted there that there was some previous studies suggesting that there might be a fiber type specific response. I think most of that was conducted in rodents. And clearly this study shows that that doesn't necessarily hold in humans. And this is important for both health and disease. So we heard a bit there about insulin sensitizing drugs being developed and glycogen storage being an important defect shown in people with type 2 diabetes. It is, of course, also important for athletes as well in recovery from exercise. So it had broad implications there. And I guess a little take home for people who are maybe starting out on their research journey. Sometimes we're disappointed when we get what we think are null findings or no differences between two treatments. But clearly here, there's a really important paper that shows that there are no differences between the two conditions here. And that's important because it actually changes our thinking based on the prior work. So it's not always a bad thing that you don't find differences between treatments. And the way to really highlight that in your manuscript is, as the authors have done here, clearly justify what the previous understanding was and how your data have ultimately altered or advanced that understanding. A couple of other things we consider are, do the methods allow an assessment that this question has been addressed appropriately? And yes, clearly they had a primary outcome that then is underpinned by some of the underlying physiology behind that. We asked the question, has the study been conducted rigorously? So there was an adequate sample size and the control measures put in place. The single leg design is a very nice, clean model. And has the manuscript been written clearly as well? And yeah, this one in particular was very clear to read. And when we're evaluating a manuscript on initial inspection, the abstract is very important. So please take care when you're submitting manuscripts to get across all of the key details in the abstract, and make sure the manuscript as a whole is easy to follow. So that's it from me. Thank you very much.
Thank you, Javier. Thank you very much, Magnus, for explaining your data. As Javier said, they're really interesting data, and how you describe your findings through the paper was really easy to follow. We're going to start with a little bit earlier, so we're 35 minutes on the webinar, which is good because I have now time to answer the questions. Okay, so I'm going to start. We have Fatima saying, commenting that very interesting experiment. Thanks for sharing. So that's a comment. But Dr. Logan Towson, which is a friend of mine, thank you very much, Logan, for being here. And so Logan congratulates you, Magnus, and Javier for the Journal Club. And I'm going to read what he asked. So I was just, Logan's word. I was just curious about your thoughts on the recent studies in rodents showing that exercise doesn't improve muscle insulin sensitivity when they're housed at thermoneutrality. So that's a very hot topic. Even though exercise obviously improves insulin sensitivity in humans, in thermoneutrality. Any idea why this might be, and what is the mean for mechanistic animal research? So it's a little bit about the translation between mice and humans and the thermoneutrality. So thank you, Logan, Magnus, or Javier.
Yeah, I'm not very confident in thermoneutrality studies, so I wouldn't have a good guess. I think, yeah. Sorry. Does Javier have any
no, sorry. Yeah, my particular expertise is human physiology. So yeah, the thermoneutrality work isn't particularly my area. Sorry.
Yeah, no, that's okay. I think we have a lot to learn about the thermoneutrality. A lot of the findings from the 90s, early to 2000s might not be replicated in thermoneutrality, which is very interesting, but somehow difficult to translate to humans. I'm going to pass to the next one. So fuel utilization in muscle will differ according to the type, duration, and intensity of exercise. So I kind of showed that my first slide with fuel utilization. And in a short time activity, it may be expected that muscle resort to glucose. So Magnus, would you expect this response to be different if exercise both were a long duration? So it's about time of your protocol.
Yeah, that is a good question. So so this experimental protocol is quite often used in the lab that I'm coming from. And so this experimental protocol contained 1 hour of one-legged exercise, but there have also been studies including 2.5 hours of the same exercise protocol, just way longer. And as I remember, the relative increase in insulin sensitization in the prior exercise leg, then it's quite similar as far as I remember. So for that sake, I would guess it would be the same or a similar response in both fiber types, because the overall insulin sensitization do not really differ, and yeah, between 1 hour exercise and 2.5 hours, as far as I remember. So I would be surprised if there was a difference in the fiber type response for the 2.5 hours protocol, which we don't see in the 1 hour. Yeah, but it's a good thought. Yeah, but on the other hand, you could say, what if the exercise bout were only 30 minutes or 15 minutes? Then I'm not sure how it would look. And then obviously it would depend on whether you would recruit all the fiber types and really put in a stress in both of them. So this experimental protocol we are using in this study is trying to recruit all of the fiber types by including some maximal, some work sessions, intensity work sessions where you work at 100% of your maximal VO2. So in that way, trying to recruit as many fibers as possible. So obviously that could be a factor if an exercise protocol didn't recruit all fiber types.
Awesome. Fatima actually made a comment that a time course response would be very interesting if participants could put so many biopsies. So yeah, that'd be really interesting.
But that would be an issue for sure.
Yeah, it's not very easy to run that study. Thank you, Fatima, and I'm going to ask Mark Turner. Thank you very much for your question, Mark. He's congratulating you, Magnus, and he was wondering if your thoughts are on the role of TBC1D1, and if you think the response would be similar to your findings in TBC1D4 in fiber types. Yeah, so this TBC1, D1, and D4 balance that we kind of don't understand properly so far, but that's the question.
Yeah, so we did actually include one phosphorylation on TBC1D4 in the paper. We haven't discussed it a lot in the results, and I didn't bring it along in this presentation. But that showed a similar regulation between the two fiber types, at least indicated similar pattern of regulation. But that was one phosphorylation site on TBC1D1, and I'm not so confident on the other phosphorylation sites on TBC1D1 in regards to insulin sensitization and whether it's involved in that.
Okay.
Do any of you have any thoughts? Are you working with TBC1D1 and TBC1D4?
Yeah, I don't know if Mark is working with TBC1D1, but we'll see. If you have any comments, Mark, please share it with me. It'll be great. So Sebastian saying that you found similar rates of glucose uptake into and glycogen formation in different types of muscle fibers. I don't know if you saw glucose uptake, I don't think so. According to textbook exercise physiology, type 1 fibers have larger glycogen stores than type 2 fibers. So does maybe the sensitizing effect of exercise last for longer in type 1 fibers compared to 2 fibers? So he's asking about the timing of insulin sensitizing effect.
Yeah, that is a good question. I would say it's not always that we find a fiber type difference in the glycogen levels between the two fiber types. So some studies have shown it a little higher. Yeah, maybe it's in type 1 fibers, but we have also found in our lab that there are no difference between the two fiber types, for instance, in this study. So it's not always there. And yeah, maybe eventually we think that we know that a large amount of the taken up glucose is going into glycogen, but it's not certain that the amount of glycogen is actually determining the degree of insulin sensitization. So there are examples where you can induce an insulin sensitization even though there's no glycogen depletion. For instance, with pharmacological activation of AMPK, it's been shown in mice, for instance, where there's no glycogen depletion, but still there is an insulin sensitizing effect. And then there's also the side of glycogen supercompensation. So where you you actually take in more glucose than you had before the exercise bout, right? And I'm not sure how that would look in the two different fiber types, but that could also suggest that the total amount of glycogen content before the exercise bout is not determining for how much insulin sensitized the specific fiber type would be.
Sure. Yeah, and what you just said has very good implications for exercise prescription for example, people with type 2 diabetes, because some people can or cannot eat before exercise. So if you have this answer that the glycogen content doesn't change the insulin exercise effect on insulin sensitivity, that's really interesting. We have another question. I'm kind of guessing that maybe Javier would like to answer that, because I think kind of falls on his expertise. Kutuba, I don't know if I'm pronouncing the name right, so I'm sorry if it's not. He's just wondering if you Magnus, had a chance to look at what happened to circulating levels of ketones and or glucagon during and after your protocol. So I know Javier studied hormones and exercise effect on that, so feel free guys to answer.
Yeah, well, I guess this relates to the model that was used in terms of a single leg model, and it may be worth discussing the benefits and limitations of that model, whereby what you get is the circulating metabolic and hormonal milieu is then similar for both limbs. And it was actually something that came up in the review of the paper. One of the questions I posed to the authors was how the single exercise leg model relates to crosstalk to the other limb. And there's some studies suggesting that with very prolonged exercise, what happens is, so you exercise one limb and that shows an insulin sensitizing effect. In other limbs, you can get desensitizing to insulin, probably due to things like increased fatty acid availability without the other muscle showing fatty acid oxidation. But yeah, maybe Magnus, if you want to comment then on the how that perhaps isn't the case with this particular study design, and any thoughts there?
Yeah, so yeah, it's actually from my lab as well. I know we have performed one study assessing this desensitization in non-exercise muscle, but that is with a much longer exercise protocol. So the one I mentioned before, 2.5 hours. I think it's a bit more difficult to actually observe an effect with only 1 hour of exercise protocol. So yeah, maybe that could talk against that. It would actually desensitize the non-exercised muscle. Yeah, and then I think the question also contained whether there was glucagon, and the levels of ketones maybe as well.
Glucagon and ketones,
yeah. So I must say I didn't participate in the human study. So that was conducted before my time in the lab. And then we just reused muscle samples from that study. So yeah, I'm not sure how the levels of different hormones actually look in this experimental model. Yeah, sorry.
That's great. But yeah, as Javier said, the mobilizing of fatty acids from adipose tissue, obviously in longer protocols, as your group has already published, probably contribute a lot of the insulin desensitizing effect on the other limb. And because one limb exercise for 1 hour, I don't know if it is strong enough to produce huge mobilization of glucagon or ketones, because they're healthy and young, but it could be the case for on the other limb for sure. Cassandra has another question. So she's asking actually about the sex differences. So she congratulates you guys for the nice work and the study. And recognizing some sex differences in muscle physiology and insulin sensitivity, would you expect any sex-specific differences if you repeated this study in women?
Yeah, that is an interesting question. I haven't really thought about that before. We don't have a lot of these experimental protocols also performed with female subjects. But I do know there's another group in America that is actually investigating us also fiber type specific differences in regards to exercise signaling. And they also include women in their studies and also have shown some examples of different responses to signaling in recovery from exercise. So yeah, you might be able to find some sex differences in regards to fiber type specific signaling. But I don't have any examples I can put up. Yeah, so I don't have a good guess on whether it would look the same or different in regards to insulin sensitization.
Yeah. So we have two more questions, but before we move to the last question, I'd like to remember all of you that we're going to continue this discussion in the network section. So Rosie will be sharing the link in the chat. She just did right now. So after we answer these two questions, Magnus, Javier, and I will go to the other room, and then we can continue the discussion. Okay? So I'm going to really quick the last two questions. The first one is David. He's asking, did you have any curiosity to investigate the group 4 protein expression in different stimuli?
So no, we didn't assess GLUT4 expression from the different stimuli. I'm not sure whether the expression actually changes much during this time course of this experimental protocol. It is only a couple of hours. Then you could assess plasma membrane bound GLUT4 maybe, but I think that analysis is a bit more tricky. So no, we haven't considered that at all now. Yeah, but I know it has been shown before or been assessed before, the GLUT4 levels between the two fiber types. I can't really remember. Oh no, yeah, maybe I can. Yeah, so I think type 1 fibers contain a bit more GLUT4 than type 2 fibers.
I think that has already been answered.
Yeah, probably.
But I think one interesting thing would be the effect of insulin or exercise, or both actually, as you did, on GLUT4 translocation to the membrane, which requires a little bit of more technique to measure that. But I think that will be better than the GLUT4 content only.
Absolutely.
But translocation to the membrane, right?
Yeah.
Yeah. The last question is from Marcos Oliveira. Marcos is congratulating you guys. And do you think you see a different pattern of TBC1B4 phosphorylation levels between the two types of muscle fibers in trainers against untrained individuals? So the training status of these people.
So I think it would be difficult to observe a fiber type difference in trained individuals because, as I remember, the overall phosphorylation of TBC1D4 is in regards to the insulin sensitizing effect or after exercise is smaller than in untrained individuals. Yeah, and so obviously that would also make it, I guess, more difficult to actually observe a fiber type difference between the two. Yeah, and then also in trained individuals, the relative effect on insulin sensitization of exercise is smaller because the general insulin sensitivity in resting muscles is increased. So the extra gap you get from exercise is smaller. So I guess it would overall make it more difficult to observe in trained individuals.
Yeah, thank you. Okay, so I think we're done with the questions and we're running out of time already. So I'd like to thank you everyone that came to see Magnus' presentation and Javier's comments. I also would like to thank you again, Magnus and Javier, for being here. It has been awesome, you guys. And Rosie again for organizing this in the Journal of Physiology and the Physiological Society. So thank you very much again. We're now moving to the other room for the network section, and I'll see you then. Okay, see you.